So this is this flowchart I was telling you about. So as the hereditary material, we're talking about it as DNA, but in some viruses it can be RNA. You can have no changes occurring, in which case nothing happens to the functioning of the organism, and we would call that a neutral situation. We can also have something known as a damage. So a damage differs from a mutation in that a damage affects the DNA in such a way that the DNA is no longer readable by DNA polymerase. So damages, if left unrepaired, by definition are lethal to the organism or the cell, because the cell can no longer replicate itself. Not because it's necessarily lost information, but because it simply can't replicate its hereditary material. However, there's a number of, and so it's hugely detrimental. It leads to dead organisms or dead cells. It is possible to repair damages, however, and we have many, many mechanisms by which we, in fact, can recognize and repair damage to DNA. That repair either can be repaired in such a way that there's no change in the sequence, in which case it's a neutral change, or it can result in change that changes, that modifies. You can have repair that modifies the sequence of nucleotides. And that would be an error-prone repair, and that leads to a mutation. So a mutation is a change in the sequence of nucleotides, and we can divide these changes into three categories. You could have changes that have the effect of modifying the functioning of the organism. So in such a way that it's not good, it results in a reduced functionality. And that is described as a decrease in fitness. This would be described as a detrimental mutation. And in fact, it can be so bad that the organism is no longer able to function. So this is not functioning because it can't replicate so much as not functioning because, not being viable, not because it can't replicate its genome, but not being viable because it lacks some crucial function. And this would be described as a lethal mutation. Alternatively, a detrimental mutation doesn't have to kill the organism, but still result in the organism having a lower amount of functionality. We have many cases where we can have mutations that don't really change the functionality. For example, if you change the codon from one codon to another that encoded the same amino acid, that might not modify the functioning of the organism. We would call that a neutral mutation. And occasionally we get mutations that, in fact, modify the functioning of the organism in such a way that the organism actually benefits from the modification. It increases its ability to survive or its ability to replicate. And we would call these beneficial mutations.